Thank you so much, Claudia, and everyone here may call me Heather. Thank you so much for the opportunity to come speak with you. Earlier today in some of our meetings, my audio went out, so I have a couple of you on task to let me know if that happens, but feel free to unmute yourself and tell me if you can't hear me so that I'm not just speaking to my cat. So today I'm going to talk to you and talk with you about social and physical environmental contributors to adverse birth outcomes and their disparities. And we'll talk about some potential mechanisms that might link environmental exposures to these outcomes. And it's a little daunting to speak with some of you. Some of you have written the papers that are the kind of back on which I've climbed to do any of the work that I've done. So I really appreciate all of your contributions to the field, and please do educate me if I'm missing really important points. I'd love to hear from you. I have no disclosures, and as mentioned, we have some funders for the work that I'll present today. And I'm starting to begin all of my talks with gratitude, because without these humans, there's no way any of this work would happen. And it's actually really fun to put this together, because I noticed a couple of things that I think might be kind of consistent with your themes based on the women I met today in all of our meetings, where this team is a majority woman effort, and yet we do have some wonderful men that join the work that we do. These humans range from students to full professors who serve as my mentors up here and help facilitate and make sure that the work happens. There are a few people from Michigan on here. At least Brisa Sanchez, I believe, was at University of Michigan before, and there are probably others. I look forward to one day getting to Michigan. So today I'm going to talk about some of the things that many of you already know, but in terms of just the raw statistics when it comes to racial disparities in birth outcomes. And I'm going to propose that environmental contributors are at least in part responsible for the observed disparities. We'll emphasize the importance of preterm birth phenotype, and don't worry, I'll talk to you about what I mean by that. And I'll share some data from some molecular epidemiologic investigations we've done to, again, link the environment to these phenotypes. And then I'm going to highlight some challenges, because you guys are all methodologic experts, at least challenges that I've observed, and maybe we can talk about ways to overcome them. So we know that non-Hispanic Black infants have over twice the risk of dying in the first year. The overall infant mortality rate in the United States is just under 6 per 1,000 live births, and we can see that it's close to 11 among Black infants and less than 5 for white infants. And this is particularly striking because Black infants just make up 15% of all births, but are responsible for, or make up 29% of all deaths. And every single of the major causes of infant mortality, there exists a racial disparity, but the most striking is that which is attributable to preterm birth or to low birth weight, with a risk ratio here of about a little over 3.5. You can see also if you count pregnancy complications, and these two are often overlapping, and it kind of depends on how we as physicians code these things on death certificates, but nonetheless, pregnancy-related complications are a big contributor to the racial disparity in preterm birth. I mean, in infant mortality. And this is and just another way of looking at the data. There are a little over 3,700 preterm-related deaths in a given year, and over half of those are among Black infants, which is striking, again, because Black infants only make up 15% of all births, whereas white infants make up 52% of all births. The reason for this is the huge disparity when it comes to preterm birth rates. And it doesn't matter whether you look at preterm birth, which is defined as less than 37 completed weeks of gestation, extremely preterm birth, less than 28 weeks. This is often the population that we're looking after in the neonatal intensive care unit. Low birth weight or very low birth weight, there are racial disparities in each of these. And you can pull these data for yourself in using CDC Wonder, but this is also the final birth data from 2018 that the CDC puts out. And previous research, although many of the colleagues of some of the folks on this call have done work beyond this. But I think when we're talking about it from a biomedical perspective, and when I speak with my obstetric colleagues and neonatology colleagues, most of the research they cite really focuses on individual risk factors. factors such as genetics or education, smoking, interpregnancy interval, prenatal care. And I'm going to talk about why each of these is insufficient to explain the population-based disparities we see. I'm going to start with genetics, which is probably one of the most controversial aspects here. So I would want to postulate this group that genetics are not the driver of racial disparities in preterm birth. We know this for a few reasons. One is that even when you look within homogeneous populations, the effect of genetic differences within populations are really, really tiny, and environmental factors kind of overwhelm the risk and play a huge role in the risk of preterm birth. And in general, when I think about health and I think about disparities in health and whether genetics might or might not be playing a role, I think about this now along a major continuum. So the continuum runs all the way from entirely genetic to entirely environmental. So the diseases that are entirely genetic with respect to their incidence or prevalence in a particular population are diseases like sickle cell disease, cystic fibrosis, or Gaucher's disease. These are diseases that are, one gene mutation and that kind of track along populations. And so the entire kind of differential risk by racial group or by ethnic group is because of this single genetic mutation that is more common in one group than another. Kind of inarguable. And so I think what happens is that a lot of physicians are trained about these diseases and then assume that any racial disparity in other outcomes is going to have a genetic component. And then that can be sometimes reinforced even with diseases that have kind of a combination of genetic as well as environmental contributions. So, we know the BRCA1/2 mutations can track along ethnic and racial identities and can therefore be more common in some groups than others. Similarly with multiple myeloma, but environment starts to creep in and have a bigger role than just in these single gene mutation diseases. And then we've got our multifactorial or heterogeneous disorders where the phenotype is really messy. And, asthma can range in its presentation from, just a little bit of wheeze to kind of severe asthma. And there can be different kind of phenotypes within asthma. Similarly with hypertension, that could be from diseases of the kidneys, or it can be due to differences in cardiovascular function. And so those phenotypes are really different. And preterm birth, I would put in this bucket. So preterm birth is a heterogeneous set of disorders that all mean that the baby was born early, but they come from very, very different distinct phenotypes. And so whenever we have these multifactorial heterogeneous disorders, the genetic contribution to them is smaller, and then even the genetic contribution to the racial disparity is almost completely nothing. Similarly, then when we get to the kind of completely environmental causes of major racial or ethnic disparities, we can think about things that are definitely not genetic at all. So trauma due to gun violence or car wrecks or work-related injuries. So if only white men are working on a fishing boat, then only white men are going to die from the fishing-related injury, right? So this is going to have nothing to do with the genetic contributions. And I would put preterm birth way over here on the environmental side. And this is also because we've got data to support that hypothesis. So this was a paper from 1997 in the New England Journal that kind of rocked the neonatology world when it came to disparities in birth weights. We've got birth weight on the x-axis. On the y-axis, we have proportion of all births. What we can see is nearly overlapping birth weight distributions between infants born to US-born white women and Black infants born to African-born Black women in the US. And then we can see a substantial shift to the left among Black infants born to US-born Black women. So there was something about being Black in America for at least a generation that was driving lower birth weights, and genetics just doesn't change that quickly. Similarly, when we look at work by Elo et al., including Jen Culhane, you can see that foreign-born Black women in the US, these are data from 27 states, have foreign-born Black women have a lower risk of preterm birth as well. So even if you don't want to study just birth weight, you want to look at preterm birth, more recent data show that foreign-born Black women have lower rates of preterm birth, and even after adjustment for multiple confounding variables. All right, so hopefully you have at least done a little bit to convince you that it's not genetic. I think then people move on to socioeconomic position or socioeconomic status, and education is a big part of that. We know that women with higher education tend to have better health in general, or humans with better education tend to have better health in general. So people will say, oh, it's the differences in education that are driving the disparity. And while it's true that within racial groups, college-educated women have lower preterm birth rates, these are data I pulled from CDC Wonder about a year ago. You can see here that women who do not complete high school have higher preterm birth rates regardless of race or ethnicity. But we can also see that college-educated Black women have higher preterm birth rates than white women who drop out of high school. And so solving education alone will not completely resolve racial disparities in preterm birth. So what about smoking? Similarly, we can see here that smokers within each group have higher rates of preterm birth. But it's important to know that fewer Black women smoke during pregnancy than white women. And so simply telling everyone to stop smoking won't resolve the racial disparity and actually could potentially widen it. And here we can also see that Black women who don't smoke have higher preterm birth rates than white smokers. So what about interpregnancy interval? I think, smoking and interpregnancy interval are two of the factors that I think physicians like to think that we can make an impact on, right? We can maybe advise people to stop smoking, or and maybe some humans might listen. Maybe we can, recommend that women don't get pregnant right away after having a baby. And indeed, there's data to support that the longer the interpregnancy interval, the less likely you are to deliver preterm, or said the other way, short interpregnancy intervals are associated with higher preterm birth risk. These numbers range from 25 to 40% depending on how short the interval is. And Black women are more likely to have a short interpregnancy interval. However, only 8% of preterm birth among Black women is attributable to short interpregnancy interval. And yes, that's a little bit more than 4% that is among white women. But even if you were to magically make everybody have a long interpregnancy interval, the risk ratio would only, between Black and white women, would only go from about 1.5 to 1.5, just a slightly smaller 1.5. And so I don't think this is going to truly make the huge dent that we would all want. The other thing that physicians love to talk about is prenatal care. Can we do better at prenatal care and prevent these disparities? And I here would say probably not. There are a couple of good studies to suggest why. So this is work from Wanda Barfield, who works at the CDC. She was a neonatology fellow about 20 years before I was in the same program. And this was her fellowship project in I think the early 90s. And she was part of the military, so she had access to this military data set. And what she said was, she hypothesized that women in the military who have universal access to prenatal care, the disparity would go away between Black women and white women, and with respect to these outcomes. And here we've got moderately low birth weight and very low birth weight as the two outcomes that she wrote about in this paper. And what you can see here is that military women, the disparities, this is a risk ratio of Black versus white. You can see that military women, perhaps the risk ratio is slightly lower than it is for civilian women. But for very low birth weight, you see only a mild attenuation of that association. And so simply just having universal access to care did not eliminate the disparity, maybe made it a little better, but didn't make it go away completely. And then in Massachusetts in 2006, while I was a fellow, we had a natural experiment. We had universal health care. This was Romney Care. This was the roadmap for Obamacare. And here I'm showing data from Massachusetts births in 2005 before universal health care. And you can see this is low birth weight rate, so less than 2,500 grams. And you can see that the risk ratio between Black and white women was about 1.6. And you can see in 2015, almost 10 years after the universal health care implementation, you can see everyone's low birth weight rate has gone down, but the risk ratio remains the same. So simply kind of improving health of everyone doesn't always get rid of the disparity. So I think this leaves us with some research gaps that a lot of epidemiologists are trying to fill, which include that knowing that individual variables are insufficient to explain the preterm birth disparities. And this was a pretty cool study that involved looking at the proportion of variables that could explain the gap. And so here, these are individual socioeconomic variables that, and this was birth in California, but can explain maybe about 17, 18% of the disparity in preterm birth. But that population variables or neighborhood level variables also explain a similar proportion. And so in order to really understand and tackle disparities, we're going to need to do more than just tackling those individual variables, the ones I mentioned before, and think about more area level variables and actions that might solve this problem. And I'm not the first person to talk about this, and I'm not going to be the last, but Ana Diaz-Rue really was among the forerunners almost 20 years ago now, where she, this is a pretty great piece for any of the students in the audience, this Neighborhoods and Health piece that was in 2001, where she put forth that residential segregation and inequality in resource distribution really changes environments and then ultimately affects health, and that these personal characteristics, these individual characteristics also interact with these factors. And some of the classic outcomes that humans have looked at when looking at these neighborhood effects of health are some of these adult outcomes, and also some childhood outcomes as well. And more recent work is focusing on preterm birth and birth outcomes. And so here we proposed also an expanded framework for preterm birth, where we think about these individual risk factors, yes, but also these macro environmental factors. And these are often the exposures to which we have less control. Some control, but less control. So the air we breathe, the water we drink, the metals in our pipes, other chemicals to which we're exposed, and then social exposures to discrimination, racism, neighborhood violence, other deprivation, etc. So, and I work with an epidemiologist named Jane Clordy at Drexel, and she's really taught me a lot. And one of her hypotheses is that these two things really interact with each other, and that our bodies can get overwhelmed. And the example that I now use is when we're studying for exams, and then we are more likely to get a cold. Our immune systems are overwhelmed. We can only handle so much. So if you have a social stressor and you add on a physical stressor, in that case the rhinovirus, you're more likely to get a cold. Here we've got a social stressor, perhaps neighborhood violence, et cetera, and you add on something like air pollution, you may be less likely to be able to handle air pollution. And so we get a piling on that happens in the United States for certain subgroups. So I'm going to first focus on air pollution. Some of you have probably done even more work than I have on air pollution, but air pollution here I'm going to talk about specifically particulate matter. So these are tiny particles. They're so small, they're less than 2.5 microns, the ones that I talked about, this PM 2.5 in diameter, and they're a lot smaller than a grain of sand. They're so small that when you breathe them in, they can directly cross over the pulmonary epithelium and end up in the circulation. And thus they can have kind of effects systemically and with among distant organs. And they work through oxidative stress and inflammation. They're implicated in a lot of these adult disorders, as well as there's data with in association with low birth weight and preterm birth. There are inequities in PM 2.5 exposure across the United States. This is another cool paper that doesn't just talk about inequity in exposure, but actually inequality in contamination or production of air pollution, such that white Americans are responsible for the larger proportion of air pollution, but Black Americans are more likely to be exposed to high levels. So there's kind of a double inequity. You can see that there's an improvement in PM 2.5 over time, and that's largely due to the Clean Air Act. So we need to be vigilant with respect to our policies to make sure this doesn't climb back up. But in general, we've gotten better, but the gap is still present. So why is it that Black Americans and other minority groups are more likely to be exposed to high levels of air pollution? Well, this is not an accident. This has occurred due to systemic racism that predates even this practice of redlining. But after the Depression, there was a program to be able to loan money to homeowners who were at risk of foreclosure. And the Homeowners Loan Corporation ended up designating neighborhoods as desirable for lending or less desirable for lending. And neighborhoods that were less desirable for lending were colored in red or redlined, and they were too risky for lending. So this is a map of Philadelphia. This is where I normally would be sitting if I were giving a lecture from my office. This is another hospital. So this is the Hospital of University of Pennsylvania in West Philadelphia. This is Pennsylvania Hospital in Center City, just south of Market Street, and this red line being Market Street. These neighborhoods were more likely to be populated by African Americans, even when you compared to other neighborhoods that had similar sociodemographics, so similar levels of education and income. These neighborhoods that were populated by Black Americans were more likely to be redlined, and thus there was less investment and gaps just widened. More likely to have industry and also roadways to go through these neighborhoods. So what we've done is, as Dr. Holtzman mentioned, is we've developed, curated a birth cohort out of the Penn Medicine birth cohort. So we call this GeoBirth. And Penn Medicine at those two hospitals that you saw as stars has about 9,000 births a year. It's majority non-Hispanic Black. It's over 50% of all the Philadelphia births. So it gets close, not perfectly population-based. But it does get close. And then we do detailed chart review of every single preterm birth that happens. And I have independent reviewers, research coordinators who read the story of each preterm delivery. And that's important because there could be misclassification of the outcome. If women come in and say, the chief complaint is contractions, then that birth, especially if it happens to be vaginal eventually, might be deemed as spontaneous. And more often than not, we'll be right, but sometimes we're wrong, because sometimes the contractions will subside, we give some IV fluids, and then the woman's found to be hypertensive, there's protein in her urine, and she's delivered for preeclampsia. That would actually be a medically indicated preterm delivery, even if a woman had an induction. And so it's important to separate those out, especially given our hypothesis that not every environmental factor will have the same impact on each of those phenotypes. We also geocode each woman's address in the year prior to delivery, and that way we can link to environmental data that varies in space as well as in time, things like air pollution and violence. So this is just some pilot data. So our first look at the GeoBirth cohort in the more recent years from one site, from the Hospital University of Pennsylvania. We can see it is majority African American, and then we can see overrepresentation consistent with national data for both spontaneous as well as medically indicated preterm delivery. These are column percent. And then we can also see that this is a majority Medicaid insured population. And preterm birth and GeoBirth looks a lot like it does nationally, with non-Hispanic Black women having a 13% rate compared to non-Hispanic white and Hispanic, Asian, and other women. And to be fair, I lump this group because the numbers are so small in Philadelphia. So unlike places like California and even in Boston, where we had more diversity, the largest groups in Penn Medicine are Black and white. So the way we model air pollution, and everyone does this a little differently, but I collaborate with an expert in this named Alan Just. He's at Mount Sinai, New York. He was actually a postdoc while I was doing my early faculty years in Boston. We were in the same research group. And he uses a machine learning algorithm to generate daily PM 2.5 exposures at a 1-by-1 kilometer grid resolution. So what his team does is they use the ground level EPA monitors as well as NASA satellite data. And then they use, it's fancier than this, but the way I think of it is kind of like leave one out cross-validation, where if you have 20 monitors in Philadelphia and you just say, okay, I know the truth at monitor 3. I'm going to pretend I don't know what monitor 3 is. I'm going to use all the other monitors, and then I'm going to use this NASA satellite data on that day to predict what monitor 3 is. And then he tunes his model, makes it better and better until he gets closer to the truth at monitor 3. And he does that for all the monitors. Then he says, okay, now I know monitor 3, but I'm going to pretend I don't know monitor 2. And he does this over and over again in an iterative way until he gets really good at predicting the truth. Once he gets really good at predicting the truth, he can then kind of give an estimate or a prediction of what the ground level air pollution was, even at some place that doesn't have a monitor. And that's how they create these daily exposures. And so when we look at Philadelphia, and you can see I've tilted the map a little bit because it's on a slightly different, this was probably hand drawn in 1937, but you can see the Delaware River coming here, Delaware River coming here. And what we can see is that this is an annual average of PM 2.5. And you can see that the highest levels of air pollution are indeed where these red line neighborhoods are. And consistent with that, non-Hispanic Black women in geobirth have higher levels of air pollution exposure here by about a half a standard deviation. Our first look was a bivariate look where we looked to say, okay, well, what about first trimester PM 2.5 exposure? Does that differ among women who deliver term versus preterm? And, at first we saw, okay, well, it looks like it's in the direction we might have expected, didn't reach statistical significance. But when we say, okay, well, what about spontaneous compared to term and medically indicated compared to term? And here we can see that the entirety of the association is among spontaneous preterm deliveries. And when we do models to adjust for this, this is the odds of preterm birth, spontaneous preterm birth, or medically indicated preterm birth compared to term for women who are above versus below the median of air pollution exposure. You can see that the entirety of this association, which once you adjust for covariates does become, quote, statistically significant, but it's all about this association with spontaneous preterm birth. And you can see that there's really nothing going on with medically indicated preterm birth. And while we adjust for race in this model, sometimes people will say, oh, well, if you have not so great overlap in your distribution of exposure, maybe you're incompletely adjusting for this, and maybe it's just still confounding. So what I like to do also is just show that this is the same if you restrict to Black women too. So air pollution is associated with, regardless of race or ethnicity, air pollution is associated with spontaneous preterm birth in this cohort. All right, let's talk about a social environmental exposure. So there are many ways to look at social environmental exposures in neighborhoods. One of them is the Neighborhood Deprivation Index, and there are many of these deprivation indices. This is Cole Brokamp's, and I like it because it's publicly available. You can download it off GitHub. It has multiple markers that he's used developed using a principal component analysis. He figured out that income below the poverty level, median household income, receiving public assistance, the proportion of humans that were uninsured, and the proportion of humans who have less than a high school education, as well as vacant housing, all were associated with hospital use in the first year of life in Cincinnati. And so a neighborhood like this, which would be right near Temple, where I went to medical school, would have a high level of neighborhood deprivation. So you see vacant housing, and then there's a high level of poverty here. These indices can be criticized, but the nice thing about them is that they decrease the dimensionality of a lot of this publicly available socioeconomic data. What we can see here is that in geobirth, neighborhood deprivation is higher among Black women on average by about 2 standard deviations compared to white women. And when we look at whether neighborhood deprivation is associated with preterm birth, in this case, it doesn't seem to matter. It's associated with all preterm birth, spontaneous preterm birth, and medically indicated preterm birth all pretty equally. And when we restrict to Black women, we see a similar association. This is per standard deviation increment of this deprivation index. So regardless of race, neighborhood deprivation is associated with preterm birth in geobirth. So what if we use a combination of exposures and say, what happens if you're exposed to both? And Philly has a nice opportunity in that there are areas that have both high pollution and high violent crime, And then other areas with more pollution, less crime, and then less pollution, more crime. And so we have this kind of ability to look at the lack of overlap. So even if we just dichotomize and look at the highest tertile of average pollution in a census tract, any given census tract, we can see that about a quarter have high exposures to both, so in the highest tertile for both. And a little under a quarter have low exposures to both. And then you can see the just single exposures in these other two boxes. What's notable, again, is that Black women in geobirth have 4 times more likely than white women to live in those high exposure categories. So high violence and high air pollution. And we can see that when you have exposure to both, your risk of preterm birth is elevated, whether you're looking at all participants or just Black women. The reason that this is not subphenotyped into spontaneous and medically indicated preterm birth is that this is from both hospitals, just 1 year worth of data, and at the time we haven't done the phenotyping at the Pennsylvania hospital. So regardless of race, ethnicity, the combination of air pollution and high crime is associated with preterm birth. So I mentioned to you earlier that we're not the only ones who have done this kind of look at neighborhood factors. And there's a group in California that's been doing this. They looked at over 1 million California births, and they did this fancy decomposition analysis to demonstrate that neighborhood matters for the racial disparity in preterm birth. So they found that they were in their models, they were able to show that they could explain about 40% of the disparity in preterm birth, the Black-white disparity. And the individual maternal socioeconomic predictors explained about 17% of this, but neighborhood predictors represented about 16%, and that air pollution might explain about 5 or 6%. What I would say is that my hypothesis is that this might be an underrepresentation. And the reason for that is that they had a little bit of imprecision on the exposure side. So they used annual zip code addresses. So these weren't a woman's own residential kind of exact address with the exposure. And so there might have been some misclassification of the exposure, more than even we certainly we suffer from that as well, but this is a little bit more extreme. And then the other thing is they didn't do any disaggregation of the preterm birth phenotype, and so it's possible that they might be able to do a little better explaining one of those two, or among even some of the sub profiles. They've got 1 million births, but they don't have access to the delivery story like we do. And so like all data sets, there are limitations, but I thought this was a really cool study using this decomposition analysis to kind of attribute these variables to the disparity. And so we're trying to do similar work, and I'm going to share some of that with you. But first, I'd like to talk about the potential mechanisms by which environmental exposures could lead to preterm birth and the associated disparities. And there are so many factors that are associated with self-identified race that may be able to, that may kind of manifest as these biologic processes that affect our epigenome, the transcriptome, the metabolome, et cetera, and then lead to preterm birth. So this would be kind of the logical way that we might think, okay, I'm exposed to air pollution, my body changes, and then I end up with a higher risk of preterm birth. And yet, what I would say is that confounding still plagues these Omics studies. And so these factors associated with self-identified race, so let's say air pollution, can affect the Omic biomarkers, but they may also affect preterm birth through other pathways. I'm going to show you kind of a way that this might be happening. So let's for a moment imagine that something like DNA methylation, one of the biomarkers, differs by race, and that it can explain a portion of the disparity in preterm birth. That's our hypothesis. So we say, okay, we know that Black women, let's say Black women have lower DNA methylation, and we think that that's mediating this race preterm birth association. That's the hypothesis. And let's say we also know that Black women have higher air pollution exposure. We showed that nationally. We're showing that in Philadelphia. I've already shown you that graph. And lastly, we know that air pollution can cause lowering of DNA methylation. And that's actually been shown in some controlled experiments where healthy humans, volunteers, gone into kind of air pollution chambers that simulate air in Beijing. And that seems like something that IRB wouldn't approve, but actually you're allowed to go to Beijing. So you're allowed to kind of sign up to go into this air chamber. And then the investigators measured DNA methylation, and they found that of some of the genes, DNA methylation was lower. So we know for a fact that Black women have higher air pollution exposure on average. We know that air pollution causes lower DNA methylation. And if we assume this DNA methylation differs by race, maybe we can get to the point where we could talk about this mediation. But there are still problems and threats. So here we can imagine that Black race is associated with increased air pollution exposure. And yes, air pollution causes lower DNA methylation, but air pollution also causes other unmeasured molecular processes that might actually be the case. And this lower DNA methylation might just be a passive biomarker that has no mechanistic role. So simply because we can do these fancy methylomic or other omic analyses doesn't mean that we know for a fact that this is along the causal pathway. Similarly, you can imagine that, let's say we know that Black race increases your risk of being exposed to racism. Racism might cause changes in your stress hormones. Stress hormones might cause changes in your DNA methylation, but stress hormones alone might actually be on the causal pathway to preterm birth or change some other mediator. And again, lower DNA methylation might simply be a biomarker, but not on the causal pathway, right? So there are other ways that you can get to preterm birth from race, even when you measure this fancy biomarker that might not be playing a role. And what I would also say is that in order to really get at this, even if you kind of say, okay, we're going to accept those threats and we'll try to figure it out anyway, you would still need representation. So I get asked this question a lot, is the reason of this, are racial disparities because of epigenetics? And the answer is we don't know. We do not know. So we just finished a systematic review of all studies of methylomics, specifically methylomic studies involved in preterm birth. And we found 16 studies that specified race and ethnicity. And then there were 9 studies that did not specify race and ethnicity at all, but we knew which countries they were from and made some estimates as to what the race ethnic breakdown is. And ultimately, only about 11% of all study participants were Black. This is not going to be enough to figure out if the difference between white and Black women is due to some epigenomic mark. So additionally, these studies suffer from some other limitations, such as being cross-sectional. So often the samples are taken at the time of birth, and so that can be caused, the differences seen between preterm and term deliveries could be due to reverse causation. So whatever caused the preterm birth, it might be causing the changes in the biomarker you're measuring. And also newborn samples might be documenting developmental stages that are normal and not pathologic. So differences between a 30-week baby and a 40-week baby might just be, if you had just taken a sample from a healthy fetus, you might have seen the same changes, or differences. And so these studies are still kind of a long way from being able to try to explain the disparities we see. So how can omics help when representation is adequate? Well, I still think they can generate hypotheses regarding mechanisms, and we can do some work to try to figure this out. So we can do some race stratified analyses, we can do causal mediation, and I'm going to show you an example of that, which is imperfect, but can still be somewhat helpful. And then mechanistic studies that, if you can show it in humans and then you can control it in animals and find the same thing, then that would be helpful. I don't do these mechanistic studies, but other folks are trying. So I'll give you an example of how we've done a causal mediation analysis. Michal Elevitz is a mentor of mine and a PI of a study, many studies, but one study in Philadelphia that she's allowed me to look at her data. They collected vaginal samples and sequenced the microbiome. And so there's, and then Jacques Ravel is her collaborator at University of Maryland, and they kind of also decrease the dimensionality of these data and put them into these community state types, so like groups of microbiomes, right? So then he numbers them from about 1 to 5. One of them, CST4, community state type 4, is particularly high risk. It's lactobacillus poor. It's dominated by anaerobic species. Claudia might say to me, you know, this is not that different from BV. I might say, yes, that's right. So this is a kind of a high-risk cervical vaginal microbiome. Whether it's truly involved in the causal pathway of preterm birth, we still don't know, but it is more common among Black women, and it's associated with spontaneous preterm birth. And my question was, well, how much could maybe the environment be affecting the vaginal microbiota? And so in this smaller cohort, we found that per standard deviation increment or increase in PM 2.5, we saw higher risk of this suboptimal microbiota, this CST4. And we restrict to Black women, we lose statistical significance, but you can see very similar effect estimates. So regardless of race, I think air pollution may be associated with this high-risk cervical vaginal microbiota. We did the same thing with neighborhood deprivation, and we can see here also that we have higher, in this case, odds, because I did multi-level modeling to cluster by census tract. But you can see that Black women and all participants, as well as Black women, have higher risk of this CST4 with higher neighborhood deprivation. And to me, this is really important because I think a lot of folks thought that, and especially on the kind of maybe the ID side or maybe on the more biomedical side, thought that this this difference in the cervical vaginal microbiota might be genetic in origin by race, and I would argue that I don't think it is. So now the question is, to what extent is the neighborhood deprivation responsible for the race microbiota relationship, okay? And so we can do this using causal mediation. So here you see neighborhood deprivation on the x-axis. So as it's going higher and higher, we go over here on the right. You can see that Black women in purple have on average a higher neighborhood deprivation. We already knew that, but we can see it visually here. These are density plots. Another way to look at this is that we now stratify by this cervical vaginal microbiota, so the high-risk one and the low-risk one, and we can see neighborhood deprivation on the y-axis. And among women with this high-risk cervical vaginal microbiota, on average, they have higher neighborhood deprivation indices. This is all participants. When we look among just Black women, we see a similar pattern, although somewhat attenuated. And then among non-Black women, we can see the similar pattern, although the numbers get smaller. When we think about causal mediation, I think the old, back in the day way to do this was this way, and I'm going to show you a fancier way in a second, but this is the old way, and this is, I think, some ways a little bit more intuitive, where you have a model where your exposure here is race, Black race compared to white race, and then your outcome is this high-risk cervical vaginal microbiota. And here you can see this, that Black women have about a two-fold higher odds of high-risk cervical vaginal microbiota. Fine, even when we adjust for a bunch of confounding variables. When we add neighborhood deprivation to this model, that is attenuated. So that gives us a hint that perhaps there is some mediation going on, but that doesn't really quantify the difference between this estimate and this estimate, and there's no kind of statistical confidence around it, and it doesn't get at some of the other things that could be either other confounding structures between the mediator and the exposure and mediator and the outcome that might be unaccounted for when we do this kind of poor woman's mediation analysis. And so Tyler Vanderweel and others have really advanced the field, and I'm sure there are maybe even people in Michigan who do this well. But anyway, I learned about this when I was in Boston. And this is to do kind of more formal causal mediation analysis that kind of allows for that confounding and interaction structure. And when we do this, and this one involves like a bootstrapping method, but nonetheless, we get this average causal mediation effect here. This is the effect of race on this high-risk cervical vaginal microbiota relationship. And then this is the direct effect once you account for the causal mediation effect of race with the microbiome. And then this is the total effect of race in the microbiome. And ultimately, we figure out that neighborhood deprivation may be mediating about 28% or about a quarter of the Black race microbiota relationship. And to me, this is really important because it highlights that race is a social construct that is a result of the environmental impacts that kind of get under the skin in some way. And so I think this supports this notion. Omics may be very helpful for us to generate hypotheses as to what's going on biomedically and they may be helpful when it comes to risk stratification. But I think we need to be really thoughtful. I think for a long time, people thought the microbiome was some kind of marker of race. And I don't think that's the case. I think it's a marker of all of the exposures that go along with being Black in America, similar to the risk of delivering early or low birth weight after a generation of being Black in America. And instead, I think we need to think about these as modifiable exposures that lead to biomarkers and outcome. And then that kind of presents a challenge. We need to mitigate those exposures and interrupt these pathologic processes. And so ultimately, we come up with a conceptual framework that starts up here with racism that leads to income inequality and residential segregation. We talked a lot about redlining. How much money we make and where we live really determines where we get educated. There are kind of ongoing educational achievement gaps. For those of you who are interested, there's a whole like school to prison pipeline literature that's worth a moment of our time. But there are big problems going on here that create quite the cycle. Racism itself can cause psychosocial stress, and then as we talked about, residential segregation can lead to disproportionate toxic environmental exposures. And when we combine stress and toxic environmental exposures, I think we overwhelm our body's systems and ability to handle these kind of external toxic exposures, and that this really is what's leading to the Black-white birth outcome disparities. So in order to have some time for talking together, I'll conclude and say that preterm birth disparities are a biosensor of racism. Measuring biomarkers that differ by race is not evidence of intrinsic genetic differences, but I do think that these biomarkers might be able to help motivate policy changes and improve physical environmental exposures. We work with folks that have really focused on green space and mitigating some of the exposures. I think there are ways to even just enforce some of the ongoing housing and water and air quality regulations that are under enforced in some cities, including Philadelphia. And that while I mentioned that education won't solve racial disparities, maybe for the individual right away, I do think that educating in communities and kind of desegregating schools and desegregating within schools can be important for not just the individual, but also for community advocacy and change. So it's not just kind of the external academic that's coming in to talk about these differences. So again, I'll conclude with my gratitude, and I am happy to have everyone unmute and talk to me. Thank you so much.
Great. Well, I just want to say that I unmute that, and I'll just for a minute appear to say that that was terrific, and it covered a lot of ground, but it really pulled things together. So I don't know if you can hear us all clapping, but thank you so much. And we will open this to questions. So just unmute yourself. I think that Heather's happy to stick around and answer some questions.
I have a question. I was wondering if you have any women in the Penn cohort who've given birth, more than one time over the time period that you've been, abstracting these records, and if so, whether you've looked at like within women variability in birth outcomes and how much there are, and if so, relating that variability to some of the social and environmental indices you've been looking at.
Yeah, so as you might imagine, so our data set starts in 2008 and goes to now. And so about in the original data set, the kind of pilot work I showed you that had about 12,000 deliveries, in that group, which was about a 3-year period, 30% of women had a subsequent delivery within that time period, which I don't think anyone would be surprised about. We do kind of cluster by woman, right? So we have the medical record numbers that we put in an indicator, so at least to statistically say we recognize the non-independence of women. So we do at least recognize that. We haven't geocoded everyone in the entire cohort. This is actually, for me, a big job because as for anyone who does EHR research, electronic health record research, it's fun, but it's messy. So we get these huge files of addresses. So I get every woman's address since she was at Penn Medicine. So this is different than when I was in Boston. They only kept the most recent address because that's where you would get your bill sent. Penn Medicine actually has an encounter address for every. Now, that doesn't mean that it couldn't be that your address is at grandma's where you get your mail, but you don't live there. That part I can't get at. But we do have an address for every encounter. So we then impute a move date from a random uniform distribution between the two encounters, right? Because we don't know exactly when you moved between those two encounters. Within pregnancy, these encounters are pretty close, so we probably have some error there. So that process of actually because about 40% of women move in pregnancy in this cohort, right? And so to generate partial exposures is one of the jobs. So that's not exactly the question you asked. But in order to be able to compare women who are not in the exact same spot or for whom their air pollution exposure might have changed between pregnancy, is definitely something that I can't wait for a student to do once our data set is clean. I think it's a great idea. I worry that we'll have small numbers, and I also worry about all the exposures that came before getting pregnant that will still be relevant for both. And so we worry, and I would love to hear from you guys what you do about adjusting for prior pregnancy outcome, because that feels to me like an over-adjustment, because a lot of the same risk factors for that first pregnancy are going to be there for the second pregnancy. So many times we'll just look, restrict to nulliparous women or pick a random pregnancy, but anyway, I'll stop.
Hi, I have a quick question. Thank you, Heather, for presenting. My first question, and I know you were moving rather quickly, but your neighborhood deprivation score, you first mentioned that you controlled for different confounders. I was just wondering what's included in the neighborhood deprivation score? How do you get that score? And are those confounders also, would you also be controlling for some of those anyway if they're included in that score?
So what we did, let me just pull it up. So first of all, definitely go take a peek at it and tell me what you think about it. This is hit the website for it, and it's pretty cool because it downloads it for every census tract in the country. But so ultimately what he did was he had a lot of these American Community Survey variables. So when you get census tract data, if you download it from the census, you get so many variables, and a lot of them are correlated with each other. And I never know which ones to use. Like, should we be using poverty? Should we be using like income for that level, whatever. So what he did was a principal component analysis to kind of try to figure out what groups seemed to be most important for the outcome that he was looking at. And so in his case, Cole was using hospital readmission and healthcare utilization in the first year of life as the outcome in Cincinnati he was most interested in. So we said, okay, which of the variables, when we look at all of them, seem to matter for that? And so these were the ones that came up. So this income below the poverty level, so how many humans have income below the poverty level in that census tract, the proportion of, or the median household income for the census tract, the proportion of people receiving public assistance, the proportion medically uninsured, and the proportion of people in that census tract with less than a high school education, and then the proportion of homes that were vacant. So these were the area level. I don't then go and adjust for more neighborhood level variables, but what we do in the multi-level model is that this is in the neighborhood part, and then the individual level variables are in the individual level part. So you could be a rich woman living in one of these highly deprived neighborhoods and still potentially have it. And once you've adjusted for all the individual level variables, the neighborhood could still really matter. It might matter differently. And so we could do, interactions or whatever to look at effect modification, but this kind of takes this as a separate thing. Other people will sometimes just use all of these variables in the neighborhood level and not necessarily put it into a score, right, or an index. So this just decreases the dimensionality. One of the criticisms of these types of analyses is that, like, this might not be the right index for Philly, and it might not be the right index for our outcome, and maybe we should be validating a local one for ourselves. And so, but there are many out there to be used. I don't know if that answers your question.
Thank you.
Dr. Burris, I was wondering if with any of the families, if like you do any follow-up, like with, like, I understand like you can't be personal with the families, but like if there's any records showing like, because like they were preterm, were there any like additional things that occurred with the child? Like were there any necessary surgeries that may have happened? I know that it's common for like children with Down syndrome that like heart surgery is pretty normal, but like hip, like there's like that. So I was wondering if that at all occurred.
Yeah, so we have a lot of different studies. This one is just of medical records, so I don't actually have any human subjects interaction with this study. The only reason it's human subjects is because we know identifiers, but we have other studies where we enroll patients, and at Penn and other places around the country, we're a neonatal research network site, so there are trials and they do long-term follow-up. But I don't particularly follow up patients for their long-term outcomes, but lots of people do, and it's what we do know is the home environment matters a lot for neurodevelopmental outcomes among preterm infants. So there's a lot of work to be done in terms of kind of making sure that we maximize our preterm infants' potential after discharge too. I have a couple friends working on that. There's a woman named Meg Parker in Boston, another woman named Susan Huang in Colorado, and they have a big multi-center study to improve kind of post-discharge care of our preterm infants. And I have encouraged them to consider some of these neighborhood level variables and neighborhood interventions to improve outcomes. It's a good question. So that's not what I've been doing recently.
Thank you, I appreciate it.
I have a question, Heather. This is really a very nice talk. I greatly enjoyed it and was informed by it. I'm wondering, you must have heard of this world that I've been a little involved in, which is the whole world of perinatal inflammation.
Yeah.
And the question I have, you've talked a little bit on vaginal microbiome a little bit. Where do you think, if it fits at all into the schema of things around prematurity, and B, if it fit, would it be part of that unexplained variance up there, the 50-60% left? Or do you think it's part of the social deprivation or even air pollution?
So is your question, do these social and, I understand, I actually think, and maybe this is not going to answer your question, but I actually think that these differences that we measure in the cervical vaginal microbiota are due to differences in immune status, to inflammation or because of inflammation. One of the reasons I think that is that we measure an innate immune marker, beta defensin, and people do all these immune profiling studies, but we see differences in those levels by these community state types too. So I'm not sure that the bugs themselves are necessarily always causing the problem. They might be just a sign of the trouble. And then that might be why when you treat BV or bacterial vaginosis, you don't see an improvement in birth outcomes.
Right.
In terms of the neonatal inflammation and like all the work that has been done by Elgan to show that inflammation is bad for our brains, it's bad probably for all of our organs, right? So I'm not sure why our brains would be the only ones affected, right? And so I remember learning about necrotizing enterocolitis or the bowel complication the preterm infants get, and learning that was bad for your neurodevelopment. And I'm thinking, how? Why? Right? And that's just because everything is systemic, right? And things are not kept in one compartment. So I don't know if that answers your question, but
No, I was asking you to speculate about these things. I know it's an interesting topic. Thank you.
Hi, my name is Zhe Hui. I want to ask a question about this neighborhood kind of experiment. You're just talking about like encouraging people to do neighborhood kind of experiment, but there are two kinds of experiment I guess we can think of. One is like the improving green space and so on. One is like, you must have heard the moving to opportunities, like moving people directly to a different neighborhood, and that changes everything, right? So I wonder, in the neighborhood kind of mechanism kind of thinking, what kind of experiment do you think would be more promising and or close to what you are studying?
Yeah, well, I think it's really interesting that some of the kind of moving to opportunity, or even we were talking earlier when I was meeting with some of the faculty around the, if there was a trial of, really doing interventions during pregnancy to kind of improve outcomes. And I think that I worry about moving people because I worry about disrupting positive social fabric as well, right? And so if you just kind of pick someone up and pluck them up and put them in a fancy neighborhood, that won't necessarily solve the problems from before pregnancy, and also it may actually disrupt some of the positive buffers that are there. So I work, as you mentioned, in green space. Sounds like you know a lot about this, but there was a trial that I was talking to the students about earlier in Philadelphia, and there's another one that's going to be happening in Louisiana, of randomizing vacant lot greening. So Charlie Brannis, who's now chair at Columbia in epidemiology, did this cluster randomized controlled trial of greening vacant lots compared to just trash cleanup compared to nothing, and found really pretty impressive improvements in mental health indicators in humans that live near the green vacant lots, using a difference in difference analysis. There was less violence in the green lots. The trash cleanup lots, not so much, not such an improvement. And so I think there is empiric evidence that, taking better care of our environment and greening might be helpful. But I think probably local improvements and regulation of industry and those kinds of things are going to be more valuable than just disrupting social fabrics, would be my gut feeling. I don't know if that helps. Oh, the dog is back. So cute.
So do we have other questions for Heather? Heather, I have a question. Are you still are you still interested in the epigenetic intermediaries, or have you given up on that?
I think it's a field that once you get too intimate with it, you start to notice the warts a little bit. So I worry about the field a lot about I worry about the cell specificity, and it's hard for me to know how generalizable some of these cheek swabs or even the vaginal swabs. We have some evidence of some markers in the vaginal or the cervical swabs that are potentially, small associations. I just worry that they won't lead to an innovation that would change how things go. But I would love to be proven wrong, and we still have some ongoing studies. I worry that I won't. It's the kind of studies that keep me up at night, because I worry that I'm measuring something that we can't act on and may not change anything. How's that for being negative, Nellie?
No, but I get it. I completely get it. Other questions?
Can I jump in? This is Claire.
Yeah.
I just kind of had a couple thoughts about the previous comments and questions about green space and interventions. And I'm not an expert on green space, and I don't know the whole field. So I'm happy to hear the findings that you said from those trials on mental health. So when I was kind of like going down this thought process, so there's also, I have a colleague here at MSU, Amber Pearson, in geography, and she is studying more like a natural experiment where they were greening vacant lots in Detroit. I think she's been stymied by COVID in terms of data collection, so she probably doesn't have results, but She and I also worked on like a secondary data analysis using birth certificate data from Michigan and measures of green space. And we have a great data set where we have linked births linked by mother over time. So we were able to look at moms who moved and moms whose level of green space changed across pregnancies. And unfortunately, we didn't find any positive relationship or any relationship between green space and preterm birth. That paper is about to come out in the International Journal of Epi. But, you know, I think that sometimes with these types of things, you know, there certainly could be impacts on mental health that we then might not see an impact on something like preterm birth that maybe has other mechanisms or pathways. But if we could, if those types of interventions can improve mental health of the whole community, that's great. And of moms, you know, that can improve other infant outcomes or the mom's postpartum health. So, you know, I'm glad to hear those. And I think I've seen, you know, more research on green space showing positive impacts on mental health or maybe physical activity, but we didn't unfortunately find, you know, any relationship, at least at the observational level with preterm birth.
Yeah, I mean, we have a proposal in to try to overlay the trial with some of our birth outcomes too, or even pregnancy conditions. You know, part of it is that I think we're going to be I mean, maybe this is also negative, Nellie, but I would expect that the impacts would be small, right? Or the associations would be subtle. And so if there's, I mean, you probably had a lot of births, so your numbers might be able to overwhelm the noise. But you know, I worry when we have a lot of noise in our data sets, and certainly I have noise in my data sets, that that we miss associations. And then the other piece is that sometimes with the birth certificate, you can get somewhat of a preterm birth phenotype, but sometimes not. So, and there's probably some misclassification, but I'd be curious, there's some evidence that walking by green space can like lower your heart rate. And so, you know, might there be an impact on cardiovascular, like more the medically indicated preterm deliveries than your spontaneous or?
Yeah, we didn't go down that path.
Yeah, yeah, right. But I think it's so good that you're going to publish the null finding, because the other piece is that maybe lots of studies are null. We don't know, right?
Yeah, I think, yeah, that's like my new niche, publishing null findings.
Oh, I have plenty. I can share them all with you. You can have your students write them up for me. Not very nice.
But thanks for your talk. It's great.
Oh, thanks for listening.
Well, I'm thrilled to learn that null findings are being published, because I keep telling my students about this bias in publication and that we need to hear more of what isn't associated, not just what is. So I'm glad to hear that, Claire. And I just, you know, I don't know if does anyone have any more questions that they want to pose to Heather?
Let me just give one little comment. When I was at Columbia, we used to call it the home of negative results. And that was largely because the Dutch famine study showed that famine in utero did not change IQ, which is a very, very, very important negative result. So I agree with you, Clay. Let's not, and Heather too, let's not dismiss those negative findings.
No, and I think Matt Gilman, who's now, you know, head of ECHO at NIH, was one of my fellowship mentors, and he told me that Heather, there's a difference between a helpful null and an unhelpful null, right? You know, so when you have really large confidence intervals and a null finding, not so helpful. But if you can narrow those and show me that for real there's nothing going on, then I'm into it, right?
Good point.
Yeah.
And if the study is well done, right? Because a not well done study of a negative is less compelling. But I just want to thank you again, because not only did you demonstrate the rigor of your research, but also the willingness to question. And sometimes we just have, you know, what we see are like a cell job, but you really laid it out there about what you thought were the problems and how you're going to tackle them. And I thought that was really great. So we appreciate that. And I do think that some of our faculty, myself included, would reach out to you and, talk about collaborations because there's a lot of overlap. So
I'd love that.
Thank
you so
you.
much. Thank you for the opportunity.
Again. Thank you again in this COVID-19 trial and error of seminars. I think it worked great, and you were our first guinea pig, so it was very successful. Thanks for spending time with our faculty and our students also. We really appreciate that.
Great. Thank you for spending your time with me.
Thank you.